A very good evening. So today's revision class will focus on biochemistry and physiology, few select topics starting with the following. If I ask you, which organ is concerned with homeostasis of glucose in body? So what would you answer? Name the organ which is concerned with the homeostasis of glucose in the body. It can be gluconeogenesis, glycogen-lysis, glycogenesis. So which organ is concerned with homeostasis of glucose? Quite simple, isn't it? So liver. Good. So you might have heard of these terms: glycogenesis, gluconeogenesis, glycogen-lysis, etc. Right? Good. Now I have a matching for you. You can find amino acids on one side and their derivatives on the other side. So try to match them based on the derivatives. So on left side you can see glycine, arginine, histidine, tryptophan, phenylalanine. And on right side you can see their corresponding derivatives. So match the following. So on right side you have tyrosine, dopa, melanin, dopamine, serotonin, histamine, nitric oxide, porphyrin, heme, glutathione. Often we see questions from these areas, so I have selected this. So these are not matching here, so all you need to do is identify the right derivative. Glycine, arginine, histidine, tryptophan, phenylalanine. Good? Yeah, see, it's very simple. See, glycine, the following are the derivatives: porphyrin, heme, glutathione. Consider this very important. Do make a note of it simultaneously. Arginine, nitric oxide. Histidine, histamine. So this is relatively easy to remember. Tryptophan, 5-hydroxy tryptamine or serotonin. And finally, phenylalanine and their derivatives, which include tyrosine, dopa, melanin and dopamine. Right? So do make a note of these points and consider this very important. Okay, so before I proceed to the next question, one of you, Ramya, says board is not clear. See, it seems to be very clear because it is streaming in HD quality for me here. So try improving the quality of your video. I'm sure you can view the board much more clearly. So is it only with Ramya or is everyone facing the same problem? Is the board not clear? Yeah. Okay, that's fine. So I think it has something to do with your streaming quality. So try to increase the definition, the quality of your video. Try HD, because sometimes if you select auto, it might take to lower resolution depending upon your internet speed, isn't it? Okay. Now let's move to the next question. What do you mean by sparing action of tyrosine on phenylalanine? Because in the previous question you have seen that tyrosine is one of the derivatives of phenylalanine. What do you mean by sparing action of tyrosine on phenylalanine? Very good, Tanya. So Tanya says when tyrosine is consumed in diet, there is no need of conversion of phenylalanine into tyrosine. Okay. Yes. So Kashish says on consuming tyrosine in diet, PA is not required. Very good. So as you know, both phenylalanine and tyrosine, both of them are aromatic amino acids, right? And among these, so based on what you said, which of them is essential amino acid and which of them is non-essential amino acid? Yes, good. Since phenylalanine leads to formation of tyrosine, tyrosine is non-essential, whereas phenylalanine is considered to be an essential amino acid. So when you consume tyrosine, there can be reduced intake or no requirement of phenylalanine in your diet. So this is called as sparing action of tyrosine on phenylalanine. Have you heard of sparing action of vitamins? Another related question pertaining to sparing action. What do you mean by sparing action of vitamins? And also we have sparing action of proteins. So we have few contexts here and there where we'll be coming across this term sparing action. Yes, phenylalanine is essential whereas tyrosine is non-essential. Very good. Vitamin C. So sparing action of vitamin C on rest of the vitamins. Vitamin C is a very powerful antioxidant. It protects and prevents other vitamins from getting oxidized like vitamin A, vitamin E and vitamin B, some of the vitamins in the B complex. So that's sparing action of vitamin C, right? And also we have something called as sparing action of proteins by carbohydrates. Usually proteins are required for bodybuilding and for growth. They should not be utilized for energy needs, right? So that's where your carbohydrates come and prevent or protect your proteins from getting metabolized for caloric needs. So that is sparing action of carbohydrates on proteins, right? So just try to have some understanding related to this. That's more than sufficient. Now coming to sparing action of tyrosine on phenylalanine, it means phenylalanine leads to formation of tyrosine. So when you take tyrosine directly, there is no requirement of phenylalanine in your diet. So that's sparing action of tyrosine on phenylalanine. I hope it's clear. Good. Now let's move on to the next question. Name the vitamin which is essential for maturation of collagen. Also, it is important for post-translational modifications. So identify or name the vitamin. So I think most of you are going with ascorbic acid or vitamin C. Very good. So vitamin C, it's one of the powerful antioxidants and it plays an important role in hydroxylation of proline, leading to formation of hydroxyproline, right? And also it plays an important role in that process, leading to maturation of collagen, okay? So vitamin C is essential for hydroxylation of proline into hydroxyproline, and this hydroxyproline can be used for estimating collagen, isn't it? So vitamin C does plays an important role in maturation of collagen. Good. Now let's move on to the next question. Often we get confused with these terms, but let's just have an overview of the same. So how do you differentiate, or what is the difference between holoenzyme, apoenzyme and coenzyme? Are they the same or is there any subtle difference? Good. So holoenzyme is the functional part of an enzyme. Ramya seems to be giving us a different equation. See, holoenzyme is the functional part of an enzyme which contains apoenzyme and coenzyme, or holoenzyme is equal to apoenzyme plus coenzyme. So apoenzyme is the protein part, whereas coenzyme is the non-protein part, non-protein organic part, isn't it? So holoenzyme is the functional enzyme, as you all rightly mentioned, and then we have the protein part and the non-protein part, which include apoenzyme and coenzyme respectively. Yes, very good. Okay, now let's move on to the next question. Give me the total number of ATP which are generated in the process of oxidation of 1 mole of glucose. So when you consume calories or in the process of burning food, per mole of glucose, how many ATP are generated? So can you give me an estimate? 32, 30, 38, 36. Okay, so just keep this in mind. How many ATP are generated in the process of, Embden-Meyerhof pathway, EMP? Eight. We're talking about total ATP production. 8 ATP in case of Embden-Meyerhof pathway. What about conversion of pyruvate to acetyl-CoA? Good. So in case of glycolysis, Embden-Meyerhof pathway in cytosol, 8 ATP. In case of conversion of, under aerobic conditions, right, you have pyruvate to acetyl-CoA conversion, 6 ATP, and then Krebs cycle or TCA cycle, 24 ATP. So total per mole of glucose, 38 ATP, right? So get familiarized with these numbers. Consider this very, very important. Okay? Yeah, if you have NADH, you can convert them into ATP, you know the conversion rate, right? Very good. Now, let's move on to the next question. What is Pasteur effect? You might have heard of it. We discussed in our E-classes as well. What do you mean by Pasteur effect? Good. Mother says inhibition of glycolysis. So inhibition of glycolysis in presence of. Excellent. Yes, Ravindra, Sumit, Monica. Inhibition of glycolysis in presence of oxygen. Why? It's because of inhibition of which of the following enzymes in the glycolysis pathway? Phosphofructokinase. And also make sure that you remember all rate limiting enzymes, key enzymes in various cycles. Consider that very important as well. Okay? Phosphofructokinase inhibition by presence of oxygen. Yes, good. Next. Okay, Davika seems to be answering the next question. What is Chargaff's rule? Also, I think you had a question on the same in the recently concluded AIMS exam. So, what is Chargaff's rule? Good. So, the number of, in a DNA molecule, the number of adenine and thymine residues are same, similar to that of number of guanine and cytosine residues. So, A plus T is equal to G plus C. Yeah, number of purines, number of pyrimidines. So, number of adenine and thymine residues are the same, AT and also GC, right? So, it's very basic, but you should never make any mistakes in these so-called basic questions, right? Okay, now let's move on to the next question. What is Marey's law? Marey apostrophe S law. What is Marey's law? It has something to do with blood pressure and another component which have a kind of relation. What is Marey's law? Yeah, pulse rate, heart rate. Good. So the blood pressure is inversely proportional to heart rate. It's because of baroreceptor reflex activity. Inverse proportional relation between the blood pressure and the heart rate. Higher the blood pressure, lower the heart rate, and vice versa, right? Yes. Good. Let's move on to the next question. In case of male reproductive system, what's the significance of Sertoli cells and Leydig cells? And under which hormones do they synthesize the materials which they are synthesizing normally? Because there was a question pertaining to Leydig cells in last year NEET as well, as far as I remember. So let's see how many of you would get this right. Good. Testosterone, Mario Cynthia. Yes, Sandeep. Spermatogenesis, Sertoli. So you can remember S and S. Sertoli cells under the influence of follicle stimulating hormone, they are associated with formation of sperms and also their maturation. Whereas Leydig cells, they are concerned with production of testosterone under the influence of LH, luteinizing hormone, right? So consider this very important and just get familiarized with these facts. Good. So even the hormones are very important. Sertoli cells, follicle stimulating hormone, Leydig cells, luteinizing hormone. Okay. Now let's move on to the next question, which is called as hunger hormone. Hunger hormone. Yeah, HUNGER, hunger hormone. Yeah, in some references it's mentioned as glucagon, in some references it's mentioned as ghrelin. I'll just write down here, GHRELIN. Good. So ghrelin is produced enteroendocrinally within the GI, especially in the stomach. It is concerned with food intake, weight, etc. It's also considered as hunger hormone. Yeah, even glucagon. Fantastic. Now let's move on to the next question, which is considered as gold standard for measuring glomerular filtration rate. The substance which is used for measuring GFR or for estimating GFR, and it's considered to be gold standard. Is it insulin? Be very careful when you're choosing options, if at all there is a question. Insulin and inulin are very close in terms of the letters, you see. Yeah, it's not insulin, it is inulin. So why inulin? Why inulin is used? So in fact it's considered as an ideal marker because of its several beneficial properties. So why is inulin preferred for measuring GFR? First of all, it is freely filtered in glomerulus, and also it is neither reabsorbed nor secreted by the tubules, and it's not metabolized or synthesized by kidneys. So because of these factors, inulin is considered as a gold standard in estimating GFR, right? So very, very important, just keep that in mind. Good, good. Okay, now let's move on to the next question. Because of time the space constraints, I couldn't add up more topics here. I still have some additional topics. We'll discuss them as well. So match the following before we proceed to the rest of the discussion. pH of secretions. You have on one column, you can see various secretions: gastric, pancreatic, bile, intestinal, and these are not corresponding pH values, but try to match them. You can see alkaline and acidic pH. So match them accordingly. Good, Monica, Tanya, Hardik. So as you know, gastric, acidic pH or very low pH, 1 to 1, 3.5, right? Low pH, very low pH. What about pancreatic or pancreatic secretions? Yes, pancreatic 8 to 8.3. What about bile? Yes, 7.8. And what about, what about intestinal? So we used to do it during our, kindergarten and high school when we used to match two columns. Yeah, very good. So do make a note of these values, memorize them, at least try to have comparative analysis as I always insist on. So gastric, you have pH of 1 to 3.5, pancreatic 8 to 8.3, bile 7.8, intestinal 7.5 to 8, right? So which has highest pH, which has lowest pH, which is close to neutral pH. Just try to have that comparative analysis, okay? Because in most of the entrances nowadays we see not just questions based on memory, but based on your analysis from your memory. Like you have these values in your mind, that's purely memory based. But if they ask you arrange the following based on the pH, like from low pH to high pH, so you need some analysis, right? So along with memory, try to have some comparative analysis always, okay? Yeah, very good. Now, let's move on to the next question. What do you mean by Herring-Brewer reflex? Herring-Brewer reflex. I don't have space here, so let me just recite orally. Herring-Breur, Herring-Brewer reflex. So what do you mean by it? Yes, vagus stretch reflex. Very good. Very good, Khyati. Yes, Kalyan. So the stretch receptors in bronchi and bronchioles, they send inhibitory signals through vagus to respiratory center when the lung is overinflated. Good. Vagus stretch reflex. Right. And what about dead space? What do you mean by dead space? How many types of dead space do you find in case of normal person, and what is the volume of dead space? Yeah, it's a Herring-Brewer reflex is a stretch reflex which is mediated through vagus. Yeah, approximately 150 ml is a dead space. And what do you mean by dead space? Yeah, we have anatomical and physiological dead space. So dead space is the area where you don't find exchange of gases. What do you mean by anatomical dead space? And what do you mean by physiologic dead space? Yeah, where you don't find gaseous exchange, around 150 ml. So from the nose to terminal bronchiole, that will be your anatomic dead space. And what about physiologic dead space? Anatomical dead space plus non-functional alveoli. Okay, I'm really sorry guys. I think there is issue with streaming. In fact, the device was overheated. I don't know how that happened. But anyways, as I said, there is no video. Let's conclude this session. But anyways, I wanted to highlight certain important points from respiratory system as well. We'll do one thing. I'll post all those important points in the description part of this video. Okay, so we'll conclude this session now. And also, I actually wanted to summarize all that we have discussed so far. But anyways, I think we tried to cover the entire video in less than 20 to 25 minutes. So let's skip that part as well. Okay, yeah, I don't know why this happens. It could be issue with YouTube app because the network seems to be fine. But anyways, I'll add additional points in the description part of the video, and it's always lovely having you all live. We'll see you again tomorrow at 9:30 Indian Standard Time in another live session. Okay, so just keep this positivity and enthusiasm going. And no matter what the challenges are, no matter what the situations are, if you are determined, there is no stopping you. Right? Okay. Tomorrow's topic, I'll let you know by tomorrow. We'll have few more basic topics like general path and microbiology. Anyways, I'll update you via our updates group. Just keep a tab on the same. And also, I heard that the admit cards are available now. So check out NBE website, log in to your credentials and download the admit card. Wish you all the best. Love you all. Okay. Good night.